Assalamualaikum and hello everyone. Today, I, Natasha Yasmin Hasnol Azahari, would like to present our research work, which is tri-enzyme system immobilized on magnetized multi-wall carbon nanotube for hydrolysis of lignocellulosic biomass. Without further delay, let's proceed with the first slide of today's presentation, where I'm going to start with a research background. By referring to the figure, we can conclude that lignocellulosic biomass is a complex substituent composed of three major components known as cellulose, hemicellulose, and lignin. The biodegradation of this component requires a series of enzymes known as cellulase, laccase, and xylanase to further hydrolyze this component into monomer sugar. These enzymes are also known as industrial enzymes as they are widely applied in industries in various sectors. Despite the tremendous application in industry, free enzymes possess poor stability and cannot be reused, which become a major obstacle to industrial implementation, leading to the immobilization of enzymes. Immobilization of enzymes offer economic properties as enzyme can be reused as well as better stability under broader working conditions. Recently, an interest in using magnetic carriers in immobilization has been extensively studied, especially since the discovery of carbon nanotubes. However, in order to establish magnetized CNT, the synthesis of iron oxide need to be done, which usually involve multiple usage of chemicals and even high working conditions that are considered as uneconomic and not eco-friendly. Besides that, the demand from the industry for establishing an economic system associated with the hydrolysis of biomass has led to the idea of single pot techniques. As most of the researchers are focusing on individual immobilized enzyme, single pot offer multi-enzyme attachment on a single carrier to perform sequential reaction. However, it is not an easy task. Thus, in this recent study, laccase, xylanase, and cellulase will be co-immobilized on magnetized multi-wall carbon nanotube synthesized using water-based system to further hydrolyze the paddy straw into fermentable sugar, as an attempt to establishing single-pot system. Research problems and scope. Previously, problems associated with native enzymes could be encountered via the immobilization of enzyme on carrier. However, concern related to the inconvenient recovery of bioconjugate when associated with insoluble materials have been brought out, leading to the implementation of the immobilization of enzyme on magnetic carrier as a solution to both problems simultaneously. As previously mentioned, the majority of researchers are concentrating on the individual immobilized enzymes. Based on the table, it can be inferred that no single pot system involving the hydrolysis of biomass using cellulase, laccase, and xylanase on magnetized multi-wall carbon nanotubes was carried out. Research objectives. The recent research objectives are as follows. Next, literature review on carbon nanotube. Carbon nanotubes can be divided into two types, single-wall and multi-wall carbon nanotubes. Due to the unique characteristic of CNT, it has been widely studied, especially from an immobilization perspective. In order to fully utilize CNT, the pristine CNT needs to undergo functionalization to increase its solubility in solvent. Among all functionalization techniques, covalent functionalization was most likely preferable, usually associated with the multiple use of acid in lab-scale procedures. Separation and recovery. Previously, separation and recovery of bioconjugate can be done via centrifugation and filtration methods. It is convenient when involving soluble substrate. However, it is inconvenient for insoluble substrate. Thus, recovery using a magnetic bar has been invented. Synthesizing iron oxide to establish magnetic carrier can be done through reserve suspension and co-precipitation method. However, this technique is uneconomic and not eco-friendly for industrial implementation. In our recent study, the synthesis of iron oxide was done using a water-based system at room temperature. Single pot study. This table represents previous successful single pot studies. By referring to the table, we can conclude that no single pot has been done on cellulase, laccase, and xylanase. Flow chart. An outline of our workflow for the recent study is provided by this flowchart. Chapter 4. Preparation of multi-wall carbon nanotube coated with magnetic nanoparticles. This figure shows the preparation of magnetized CNT using newly developed water-based techniques. As shown in the figure, deionized water was used as a solution medium instead of chemicals, and it was successfully done at room temperature conditions. FTIR analysis was performed on pristine, functionalized and magnetized CNT to further validate the efficiency of a water-based system in establishing magnetized CNT. As shown in the figure, a distinctive peak of C double bond C can be seen on pristine CNT, indicates the graphite structure, while additional peak of C double bond O and CO bond can be observed on functionalized CNT, indicating pristine CNT was successfully functionalized. After the synthesis of iron oxide using water-based system, the formation of new functional group FeO stretching validates the efficiency of the water-based system in establishing magnetic carriers. Apart from that, SEM analysis was performed to further study the morphology of CNT. In contrast to p-MWCNT and f-MWCNT, m-MWCNT appear to be spherical in shape, coarser, with random agglomeration resulting from iron oxide attachment. Besides that, through EDX analysis, an additional of Fe element on m-MWCNT supports our conclusion that using water as a solution medium can lead to the formation of magnetized multi-wall carbon nanotube. This figure represents untreated, dilute acid pre-treated and biologically pre-treated paddy straw using single pot analysis. By referring to the figure, the degradation of lignin is confirmed by the decrease in absorption depth at 2921, indicates CH stretching. Besides that, according to previous study, the prominent peak in treated paddy straw was assigned at peak 1515 and 1647, which indicate lignin removal. Thus, it can be deduced that lignin successfully degraded in both paddy straw sample. Through SEM analysis, untreated paddy straw illustrate as rigid, compact, smooth, and highly oriented structure. Following pretreatment with dilute acid and laccase, the pretreated paddy straw surface morphology changed to become disorganized, porous, and irregular. For biological pretreatment, which is using laccase, dumbbell-shaped silica bodies can be observed, indicating destruction of microfibrils. Next, reusability of single pot treatment on paddy straw. By referring to the figure, as the number of cycles increase, the reducing sugar decrease might be due to several possible reasons. First, denaturation of laccase enzyme. Second, insufficient laccase concentration. Third, detachment of enzyme from carrier. By comparing untreated and dilute acid pre-treated paddy straw, untreated paddy straw secrete higher glucose and xylose due to the specific reaction of laccase enzyme to disrupt the cell wall structure compared to random attack on paddy straw structure through acid treatment. Conclusion. As a conclusion, we conclude that single pot system consisting of tri-enzyme immobilized on magnetized multi-wall carbon nanotubes synthesized using water-based system do show reusability properties even though the number of the cycle is not that high. Publication. The publication of our previous research work are as follows. That's all from me. Thank you.